Hi, everyone. It's 10 o'clock, so let's get started with this external webinar. In today's presentation, we have Steve Quinn. He's an associate professor of biostatistics and bioinformatics at Emory University, and he also obtained his PhD degree in statistics at the University of Michigan, and then went to a postdoc training at Harvard University. His current research focuses on biological big data integration and mining. And in today's presentation, he's going to talk about Omicseq, genomics big data search engine, and knowledge discovery system. So welcome, Steve Quinn.
Thank you very much. Good morning and good afternoon, everyone. And thank you for coming. It's a great pleasure to present our work at this webinar series. And I'm a big fan of BD2K and bioCADDIE in particular. And many of our ideas behind our Omicseq tools are in line with the philosophy of bioCADDIE. In particular, I like this quote in the overview page of bioCADDIE website, that bioCADDIE wishes to do for data what PubMed and PubMed Central did for the literature. So PubMed, as we know, is a great bioinformatics resource. I still remember about 14 years ago, when I first entered this field, the first website I was told about is PubMed, and it has been my web browser start page ever since. In my opinion, the best feature of PubMed is that one can use it to look for articles in pretty much all the journals in the medical field. Without PubMed, you have to go to the individual journal website to search for an article of your interest, and which can be a huge hassle. And up until now, I think it's safe to say that literature has been the major, if not the only resource for biomedical knowledge. And these are for good reasons, because with peer review, the information contained in the literature is accurate, and since it is expressed in language, it is specific and definitive. And it is easy to work with, because all the infrastructure has been built around it. Like PubMed and MASH, they're already in place. And over the last 30 or 40 years, there are powerful natural language processing technology that have been developed, which can be really useful for effective literature mining. However, I would argue that literature has its own limitations. And here are several reasons. First of all, the knowledge is not distributed evenly in the literature. For obscure gene, little information can be found in the literature. So we have done a simple survey that we queried all the every gene on the human genome against PubMed, and we plot the distribution of number of papers we found. As you can expect, the distribution is extremely skewed. Here in this plot, I show the x-axis is log transform. So from this plot, one can see that more than 30% of the gene in the genome were not mentioned in any papers. So there's a huge bar to the very left. So there's zero record in the PubMed. And the median of the distribution is only 5, which means 50% of the genes in the genome were mentioned in less than 5 papers. And second, and only quote-unquote interesting and significant findings were reported, because the space is limited. So the mundane events like the most TS binding and gene expression changes do not make to the papers. And because of the publication is polished, and so the content can sometimes be subjective and selective. So in a word, it's a bias. And I think not all the biomedical knowledge is in the literature. And with the development of all the high-throughput technologies, I think there's a lot of questions are actually can be found elsewhere. Like the questions, for example, questions like which transcription factors bind to the promoter of my favorite genes, like which facial mark is the most enriched in the genomic locus, and does the expression change in prostate tumor versus normal, and does the locus belong to any DMR, and is SNP located in enhancers or CG island or also conserved regions. So this cannot be easily found in the literature. So where are this knowledge? The answers, my friend, are in the data. With the development of high-throughput technologies like next-generation sequencing, we are blessed with tons of genome-wide profiling data, or some people call it omics data. It is not uncommon that a paper is accompanied by tens or sometimes hundreds of data sets. The data set can be ChIP-seq data, RNA-seq data, isomi sequencing data. So I would argue that there's more information contained in the data than actually in the paper. However, the data is difficult to deal with. The bottom line is that there's no infrastructure like PubMed, so you're on your own. For most of the biomedical researchers, we don't have the training, skill, resource, and time to do this. So that is a real serious problem. So to use the data, we have to locate the data, we have to download the data, we have to process data. And each step is non-trivial, time-consuming, error-prone, and not reproducible, and need professional assistance. So even though there are massive data that have been generated, so very few people, and most of them are bioinformaticians, is actually using it. But the majority of biomedical researchers, they don't even know that this data existed. So there's several problems or challenges in the biomedical field right now. So first of all, the data are scattered around, and it's difficult to locate and relay them. So people like to say that they're in silos. Another question reminds me of this. So this is a catalog from IKEA 2013. And what you see are beautiful furniture. They're modern, they're comfortable, and best of all, they're inexpensive. But don't let these pretty pictures mislead you. Because when you walk into an IKEA store to buy this furniture, what you'll find is actually this. So it's difficult to actually take a lot of work to assemble this furniture into the beautiful picture you saw in the previous page. And not sure about you, but I can tell you that this is really a difficult task. So I feel that we have the same problem in our field, because when you turn the raw data into a final result, we need to do processing, we need to do quality control, we need to do normalization, and all of these jobs are more complex than assemble furniture. Right? So although these data are available, much work is required before they can be used by biologists or clinicians. So this is the current state in our field. So what to do with that? So we have a dream that one day there's a website that I can go to that can give me links to all the biomedical data that I ever produced, just like PubMed did for literature. That one day there's a database that stores data that are processed and ready to use. That one day there's a search engine from which I can get information on pretty much any gene or any genomic entity instantly. And currently, there's no resource available to do this, to provide this service to us. So we began to working toward this dream. So we started the Omicseq project. And at the start, we began from genomics data. So these are Excel spreadsheets. So from my collaboration with biologists, I found that my biological friend, I really feel comfortable working with Excel spreadsheet. And this is pretty obvious because you have a very simple structure. So typically, each row represents a feature in the genome, like a gene. And each column is a data set. And this can be anything, can be a p-values, can be a log intensity value from microarray, can be RPKM from RNA-seq data. So with this data in Excel spreadsheet, they can do a lot of things. They can compare results from a different experiment. They can take fold change. They can do t-tests, and they can do clustering. They can do classifications. So they can do a lot of things. So then we said to ourselves, isn't it great if we can put all the data, all the genomic data available to a single giant Excel spreadsheet? So if we can do that, then imagine this Excel spreadsheet will have 30,000 rows, and each row represents a gene in the genome. However, it will have a zillion columns in this data set, because all the data sets will be there. And some column will be ChIP-seq data, some column will be RNA-seq data, and some column will be microarray data. So if we can do that, then this really solves the two major problems we mentioned earlier. First of all the data are now in one place. And second of all, they are processed and they're ready to use. So this will be really cool to have that. And for the type of data, and there are actually two major categories we can put into this this Excel spreadsheet. The first categories are data that directly from the experimental assays, like IPKM values from RNA-seq, like rate count of promoter from ChIP-seq, like a log intensity value from microarray, like a pulsing index from growth-seq, or average methylation level and promoter from BS-seq. But this is not the only data you can put in there. There's some other quantitative attributes from summary statistics, sometimes from multiple experiments, or from algorithms, or from a special type of measure. The example in this category is like a proportion of patients in a disease cohort that have mutations, p-values from detecting differentially expressed genes, free energy from RNA folding algorithms, a degree of centrality in protein-protein interaction network, which are very important for system biologists, or a C-index from survival analysis when you have clinical data. So there's many sources that can generate this data. We can find data from there. So there's a public genomic data repositories, like GEO, SRA, and Express, and then there's a disease-specific repositories that are publicly available. Like for cancer, there's the TGA, there's FEGC, CCLE, and there's also large consortium, often international consortium, that are tasked to generating those very useful resources like ENCODE, epigenome roadmap, and ModENCODE, and DTAC. And then so all of this data can be added to our Excel spreadsheet or Omicseq. And also we want to plead to the community that please send us your data to be included in Omicseq. It doesn't matter if it's published or unpublished. And we'll make sure you get credit when your data really help the researcher. And sometimes we also welcome the unique data types that are currently, maybe some of your data is currently not available in our current database, and we also welcome that. And this doesn't have to be directly from the instrument. It can be anything like gene-based scores, like summary statistics, like p-values, q-values, some in-tests or some scores. Everything as long as they're quantitative, as long as they're genome-wide profiles, and they can be fitting nicely into our database. So obviously, we cannot use Excel to develop this resource, and even Access won't do this. So we really need a database. So how to store this data? So the characteristic about our database that we have a moderate number of IDs. Like there's less than 30,000 genes in the genome, but we do have a massive number of columns. Another key feature is that we need to update this database pretty frequently, because the whole community keep generating new data, and there are almost a lot of new data generated almost daily. So we need to update our database pretty frequently. And because of this, the traditional classical MySQL database cannot do this. It does not meet our demand. And we really need a NoSQL database. And currently, we're using MongoDB in our in Omicseq. And from our experience so far, it works very well. And suppose we have collected this large amount of data, then what to do with it? How can we utilize this data? So currently, maybe it's obvious to you that the query, the data set will be a very useful features to have. So existing data set querying functions only use metadata. For example, they can find the data from the cell type of interest, from the experimental types, or from certain tissues. However, they cannot go into the data set. That is, it is highly desirable if we can query the content of data set directly, like a gene in the genome, or genomic regions, or microRNA, or certain pathways. So it will be nice if we can provide that feature, just like we showed in this page. Just like an internet search engine, if we can enter a gene, then to search this database, that would be very nice to have. So to be able to do that, we really need to prioritize among all the data sets. That is, given a gene, we want to display only those data sets that this query gene is playing quote-unquote important roles. So this is similar to the internet search engine, right? Given a search term, we want to, what web page do we show at the top? What web page do we show on the first page of this result page? So the key challenge here is how to compare two data sets that are of different types. So to do that, we have a hypothesis that a gene, if plays an important role in a data set, then its score will rank at the top among all the genes in the genome. So based on this hypothesis, we developed this so-called track rank algorithm to rank data set using this idea. So the algorithm works like this. At the beginning, we have a raw data from this experiment. Then we do processing normalization and then summarize this raw data into a summary statistic or score. So this score is a value for each gene. Then with this summary statistic or scores, we can rank the genes in the genome from the top to the bottom. Then we can rank the data set using this rank. So here is a simple illustration of this idea. So suppose we have 5 different data sets, ChIP-seq data, RNA-seq data, growth-seq data, bisulfite sequencing data, and CLIP-seq data. And suppose we're interested in gene A. We want to query this gene. Then we can find out how this gene A ranked in each of these 5 different data sets. Let's say in ChIP-seq data, gene A, the number of reads in the promoter region of gene A rank 1,500 among all the genes in the genome. For RNA-seq, gene A ranked near the top, number 5 among all the genes. For growth-seq, rank is 800. For bi-seq is ranked close to the bottom, 20,000. For CLIP-seq, the rank is 80. Then, given this rank, we can determine the orders. And because in the RNA-seq data, this gene A ranks very high, number 5. So it's natural to put this gene first. Then the next one is the CLIP-seq because it ranks 80th. It's like a quarter of 1 percentile. So we run this gene next. For the remainders, run 800, 1,500, and 20,000 because they're pretty in the middle or near the bottom. So probably we're not going to be interested in that. So they will be ranked lower. So by doing this, we'll be able to prioritize this among this data set, even across different experimental types. So here I showed you is a typical Omicseq result page. At the top is the search box. So you can see the interface is actually very simple, just like an internet search engine. In the search box, you can type in any gene names. So for example, here we search ERG, which is important in prostate cancer. And then here are the results. Each line represents a data set. The first column is the ranking, and the second column is the data set ID, which we assign. And in the future, this can be the DDI. And the data type tells us that this data set is CNV data. And the sample column tells us this data is from a TDGA, from a leukemia patient. And the next column tells us this is the tissue types and also the status of the sample, and also if there's any transcription factor involved. And the order tells us this gene, ranked number 12 among all the 22,000 genes in the genome. And the percentile is 0.054. And the data is coming from a TDGA study, and also if we know the source of the lab, we also include it there. And there's some more information you can explore. For example, the metadata contain the information about this patient, and there's a PubMed which linked to the paper talking about this data set, and then there's a geo link if you want interested to download the data. So each line represents a data set in our database, and these are ranked by these orders. And behind this Omicseq web page, there's a database and infrastructure. And here is the illustration of the underlying infrastructure. To the left, this is the genomic data which live on the internet. And we have on our system, we have a data crawler, and periodically go to the different public repository to grab the data of interest. And then this raw data will be processed locally into score and then into ranks. And then this rank will be stored in the database, and also the metadata will be generated and stored in the database. And to the right is the query part of our tool. There's a web portal, there's a query job management system to facilitate the querying submitted by the users. So we have presented our work to several groups, and we have some common feedback from the audience. So one concern is about the normalization. So we realized that normalization is an important issue. But in fact, what the TrackRun did is exactly a normalization procedure, because all the data sets, doesn't matter what types they are, and they are normalized to a percentile. And the percentile is a value from 0 to 1. So data from different types, they are all transformed into a percentile between 0 and 1, such that they can be comparable. So in a sense, we think this is a normalization, the best normalization one can do. And also, people have questions about metadata. So by default, metadata is not involved in the ranking. So we only compare the numerical value among the genes to determine who ranks higher. And then in the second step, among those highly ranked genes, then we can use the metadata to tell the user the potential feature, potential function of this gene. However, if the user is interested, then the metadata can be considered. For example, if the user is only interested in a certain subset of data, like a certain disease, a certain cell type, or certain experimental type, then this metadata information can be added to the query. So this is like an advanced search in PubMed. So user can add an additional attribute from the metadata into the search. Then the metadata information can be used in the search. And so what we did is only a very simple method, and this by no means this will be the final method. And there's some investigators in bioCADDIE and also some other places. They have a lot of experience working with the metadata. So we really like to work with you to develop more sophisticated algorithms that combine the numerical data and the metadata to improve the ranking mechanism. So this is certainly something we can work together and to improve on that. And then there's also some question about the QC issues. And the QC, again, is very important. Our philosophy toward that is we're not going to remove the data just because some people question about the quality of this. Instead, what we're trying to do is we're going to provide some basic QC measures so that the user can decide whether they consider this data set or not. And there's a lot of QC measures that can be informative on that. For example, there's a total number of reads, there's a read length, there's average breadth scores from the sequencing experiment, there's a mapping rate, there's duplication rate, and we'll try to provide those information as much as possible. So users can determine whether they are happy with the quality of a particular data set. They can even set up filters, for example, to remove all the data sets that have less than 10 million reads, things like that. So that's one thing we can do. But obviously, we recognize that the quality issue is complex. There's many factors that cannot be simply determined by a few measurements. Then, in order to improve the data quality assessment, we give the power to the user. So we resort to crowdsourcing. So what we plan to do is to provide a rating system, like Amazon did, a 5-star rating from 1 star to 5 star. And the user, if they like the data set and they think the quality is good, they can give a very high, like 5 stars. If they have some question about this, the quality of the data set, they can give a lower rating. So the future users can benefit from the previous judgment, so they can make their own decisions whether to trust the quality of the data or do some additional analysis in order to feel more comfortable with the quality. In addition to the ratings, we're also going to provide a common box. That is, in addition to this categorical star ratings, and the user can actually leave notes on the quality of the data. And for example, if they don't like a particular aspect of the data, they can say that. So in the future, when another user come across this data, they can read the previous comment and try to understand what are the previous concerns about this particular data set. Then he or she can make her own judgment whether this quality issue is serious enough to prevent her to leave this data out for consideration. So we're basically going to resort a crowdsourcing mechanism to improve the quality assessment for the data set in our database. And so when my vendor, suppose we have this resource built up, we have this huge database, and we also have this query functionalities in place. So what can we use about it? How can this tool benefit your research? So we have to really think hard about this, and we believe there's many scenarios that the user can benefit from using Omicseq. Here are just some examples. Like for example, you can use Omicseq to explore your favorite genes. You can use that to annotate unknown genes. You can use Omicseq to put your result in context. You can compare genes. You can also use Omicseq to find genes of similar function. Here, I want to elaborate a little bit more on the first two scenarios. In the first case, we know that every biologist have their own favorite genes. So they have studied for a long time. They know very well about these gene functions and what their interesting features are. But by conducting this Omicseq query, then because we have such a diverse collection of data sets, currently in our database we have more than 50,000 data sets from all different fields, including population genetics, evolution, cancer genomics, and you name it. So the chances are the researcher might find something that they're really interested in, but never know existed. For example, this scenario could happen to a researcher that among the top ranked data sets, it turns out CTCF binds to its promoter, which is ranked within the top 50 in the entire genome. Or it's possible that he or she found out that 5% of mutation rate of this gene was found in a cohort of head and neck cancer patients. And then this actually ranked this gene one of the top 20 most mutated genes. So this fact, because there are so many such facts out there, so one might not know to all of them. So by doing this simple query, you might find something that you don't know personally, and you might be interested. So you might then end up digging deeper on this data set and to learn something new about your favorite gene. In the opposite case, suppose you come up with an unknown gene. You can also use Omicseq to learn something about it. So this is like a background check. Like consider a scenario, like for example, if you do a GWAS study, then it turns out there's a significant SNPs have a very significant p-values that are located near a gene. And then chances are this gene are new to you, so you don't know about the function of this gene. So the first thing you can do is go to PubMed to see what people have been saying about this gene. But like I mentioned, 50% of the genes have less than 5 papers talking about it, so chances are you won't get much information about this gene from PubMed. Then as an alternative, you can come to Omicseq to learn more about this gene. Because when you do a simple query, you may find out in which data set and in which conditions that this gene has unusual behavior compared to the other genes in the genome. For example, high expression, or differential methylation status, or TS binding, things like that. Then when you find this out, you can check the study, check the publication for more details. So maybe from there, you can find clues to link this gene to the phenotype of interest. In other user cases, and then suppose we have several genes we don't know their function about. What we can do is we can do a query of each of these genes, and then we can collect the top ranked data set. Suppose set a threshold, and we can identify the top ranked data set, and then we can collect their metadata and look for enriched keywords among this metadata. So we may be able to find some clues about this gene's functions using this method. So there's many different ways that you can utilize this resource to benefit of your research. And currently, Omicseq only contain human genes, and all the data is arranged based on genes. However, this framework can easily be extended to other scenarios. Like for example, for human, not only for every genes, we can collect data based on exons. We can also collect data based on microRNAs or based on same variants. We can use the same idea to do the ranking and to do the querying. And this doesn't have to be just for human species. It can be applied to other model organisms, like mouse, like yeast, like plant. And then better yet, this data can be connected. For example, when you query a human gene, not only you can look for the interesting data sets that are in the human database, you can also identify the ocelot genes in mouse and search the mouse database. And in the future, Omicseq will facilitate this functionality to allow cross-species queries. So just like everything, Omicseq also have limitations. And the primary limitation we can think of is because the biomedical data are pretty diverse. Their types are very different. Not all the data types are suitable for the current Omicseq setup. For example, gene interaction data is unable to fit into the current Omicseq setting. Also, the imaging data from pathology studies, they're not yet be able to fit in. And in more general, like clinical data, they're not able to fit in yet. Also, any categorical data currently, unless it's very close to continuous, they're unable to be accommodated into an Omicseq database. And another limitation is because our query are purely data-driven, so the result may be spurious. However, this problem exists for every big data analysis, because unlike from the literature, you can find causal relationships. In big data analysis, all you can find is the association relationships. So the expert need to be consulted in order to determine whether these associations are genuine or not, and whether the association can be lead to a causal relationship inference. So this needs to be treated carefully. However, we think this is a tolerable problem because more hypotheses will be generated from this analysis, and more knowledge can potentially be discovered. And there's also some technical challenges that we may face. This might primarily coming from the IT side, because you can imagine when the database increase, then there will be some IT issues to be needed to resolve. However, the biomedical field is not the only field that faces a big data challenge. We can learn some lessons from other disciplines like high energy physics, like environmental studies, and even people like Amazon. For future work, we're going to expand Omicseq in the following areas. For content, we're going to and we're ready to add more data to the database and more different diverse data types and from more different species. And also we're going to do a better job to standardize and include more richer metadata. And we're going to create a database for microRNAs, for SNP variants, and for genomic region to facilitate query of different types. And we're also interested to add a functionality to that, like data mining capabilities, and recommendation system, and visualization. And investigator, like bioCADDIE investigator, have a lot of experience in these areas. So we really like to work with you to develop this facility capability to make better use of our resource. And last but not least, we'd also like to improve on the methodology side, working with statisticians and data scientists to develop a novel statistical learning method and a big data analytical method to better use this resource. So let's work together. So we're really looking for academic and industrial partners to develop Omicseq together. So currently we don't have any funding for this project yet. So we'd like to work with you to make it sustainable, to develop, make it. We believe this will be an incredible resource for the community. So we'd really like to work with you to continue to develop this resource. And to the data generators, we want to work with you to host your data to make it more accessible to the whole communities. So it doesn't matter if your data is processed or not. It will be great if it's processed, we can easily add it to our database. If it's not, then we can subject it to our standardized pipeline and to convert it into processed data and add it to our database. And to those end users, like medical researchers or clinicians, we'd really like to listen to you, to hear your opinion on the like which function you like, which function you don't like, and which new function you would like to be added. So we can work together to make this resource more useful for the communities. We also like to work with implementations to develop tools and functionalities on top of this Omicseq resource, and to develop the recommendation system, to using the user histories to make the resource more user-friendly to the wider communities. Also, we like to work with statisticians and data scientists and to develop novel analytical methodologies. Because currently, we don't have a real big data, even though a lot of people are talking about big data and analytics, but we don't have opportunities to work with the real big data. And by developing Omicseq, we believe that we'll present a true, real biomedical big data. And so that this can benefit to the statisticians and data scientists working in this area to develop better technology to analyze this type of data. And in summary, our ultimate goal in Omicseq is trying to explore big data to complement and assist traditional hypothesis-driven biomedical research. We believe to do that, we really need to build the infrastructure at the first stage. We need to collect data, we need to format data, we need to develop tools. So by doing that, we'll be able to give biomedical researchers access and analytical capabilities to handle biomedical big data. So before close, I'd like to offer some final thoughts. And in our opinion, we think that for classical hypothesis-driven biomedical research, and they typically conduct experiments and do data analysis on a fixed plan. So they have a pretty good idea which data they're going to generate and which data they're going to analyze. So for them, I don't think big data can be really helpful to them. So the BD2K is quite different. In my opinion, BD2K is to discover something unexpected, unsolicited. Without this big data infrastructure, we won't be able to identify that. So this is like let the data guide you to something you never know existed, to find something serendipitously. So this practice is really generate rather than prove hypothesis. So we think this is quite different from the previous biomedical research. So they have a pretty promising and will, in our opinion, will revolutionize biomedical research field. However, in order to do that, the really important work is to really have a new infrastructure to do that, to facilitate BD2K. Just like I showed you the quote from the first page of my slide, we need to build something similar to PubMed to be able to facilitate this BD2K discovery in a new age. So I'd like to acknowledge my coworkers. This is a very ambitious project. A lot of people are involved. I want to thank Dr. Fusheng Wang, who's an assistant professor in Emory Biomedical Informatics, who's an expert in database. I want to thank Steve Pitar, who is a data scientist in biostatistics and biomedical department. We work together a lot. And Tianlei Xu, Xiaobo Shen, Man Wang, Zhang Qin Shen, and Andy Wu are graduate students working on this project. We also have a IT team back in China led by Bai Wu, and they spend a lot of work to optimize and develop this database and infrastructure. And we also want to thank Kevin Lei, Tanya, Taswang, and Randy Asset in Emory for technology assistance. And with that, thank you very much. And we have actually a YouTube video to show a very short video to show you the basic functionalities of Omicseq. You're welcome to check it out. And you also contact us. Here's my contact information. Please call me or email me. I can give you a more live demo to show you the functionalities of the current system. And I want to welcome any feedback you can send us, and we can work together to make this resource a better one for the whole community. Thank you very much.
The conference has been unmuted.
Any questions for Dr. Quinn?
Yeah, can you guys hear me?
Yes.
Yes.
Yeah, this is Jim Zhang from UT Health. Steve, I just want to ask you one question to clarify your statement. You said on average, the gene-linked PubMed is is around 5, the median.
Yes.
Is that for all the genes, for all species, or are you looking into different organisms?
Just human.
Just human.
Yeah.
Where did you get that information?
We did it ourselves. We have
Oh. Okay.
We write a script for it systematically. Yeah.
Okay. So. Okay. So there is a gene to PubMed file from NCBI. I'm not sure. Are you aware of that?
No.
So if you look into that file, I think the actual number of PubMed abstracts linked to the gene is much higher.
We certainly acknowledge our query is very simple-minded, so we're not surprised to see there's more paper about a single gene. But the message, I think, remains the same. For many genes, there's little information you can find from the literature, and that's a fact.
Yeah, that's certainly true for some genes, but I'm just afraid that the statement you make it's kind of a little bit not accurate because a lot of study relies on this information. Like we use this information to develop some methods. And, by our account, based on that information from NCBI, the number of abstracts linked to, genes is much higher than what you stated. I just want to clarify that. Maybe I can send you the link about that gene to PubMed file. Maybe you can take a look.
Yes, please. Thank you.
Any other questions for Dr. Quinn? So I have one. So you talk about for future work that you are planning to expand use of your technology for other types of data, such as clinical data or imaging or pathology. Have you thought, are you planning to use the same Omicseq technology, the same infrastructure, or you're planning to develop a different tool in order to look into other types of data?
We are, I think some new technology has to be developed. The current setup doesn't fit in with those imaging data. However, a portion of those data, like clinical data, can be added to it. So this will be explored in the future.
Okay, thank you.
I had a question too. This is Susanna Santura from Oxford. I noticed you said you use the metadata more for advanced searches, if I got it right. And I saw when you had the screenshot, you had an example of the metadata. I think you had the tissue, the factor. And I wonder, because all the metadata is very inconsistent across all the different data sources that you take data from, inconsistent in the level of what kind of metadata is recorded, but also what kind of terminology is used to annotate that metadata type. How actually do you harmonize this? Do you have an internal ontology that you map everything against?
We don't. We have actually, current stage, a very simple method to add the metadata. We basically copy the format used in PubMed. So there's an advanced search capability in PubMed. So you can specify the journal name, the author name, the institution, things like that. So in our case, we can allow users to specify a cell type, a disease, or a study, or experimental type, things like of that nature. So it's not really sophisticated, but I think it's useful for many of the researchers.
So I'm sorry, just to clarify, you allow them to specify their own, sample type issue, but you don't enforce any terminology on your site, and you don't map the different value for the different fields. Because what I'm trying to say, these are not standardized.
Right. I agree. So basically, what we consider right now is a pretty simple, I would say, metadata, like cell type, like disease, like experimental types, like labs, things like that. So we certainly don't have a very sophisticated metadata analysis capability right now. We don't have ontology at this moment. But certainly that's something I would like to have, and I would like to work with people who have experience in this respect to improve these capabilities.
Okay, thank you.
So this is I was wondering, have you considered putting together the papers from NCBI gene search tool to be integrated with Omicseq? So when people search a given gene name, you can see both the paper and the data set side by side. So it's easy to find some linkage or find new findings.
Yeah, so that's a very good suggestion. I really like this idea. So currently on our result page, we have a link out to PubMed. So you can open a new window and to search the gene in PubMed. And certainly we can do more than that. So one idea we have is that we can develop a word cloud from the literature side and to incorporate the information collected from the literature into the result we collected from the data. So certainly we will be interested to pursue that direction. And I think that's a great suggestion. Thank you.
Any other questions for Dr. Quinn? Okay, so I think we can end this presentation. Thank you, Dr. Quinn, for this very relevant presentation for data index and discovery. We're going to have the next external webinar in January 8th from 10 to 11. So I'll see you all then. Thank you.
Thank you very much.
Thank you.